Alright, so today we will be talking about stages in beta oxidation of fatty acid. Stages in beta oxidation of fatty acid. So after you've given that brief introduction of fatty acid, that on beta oxidation of fatty acid, that it is actually the breakdown of fatty acid at the beta carbon there. And so given that as an introduction, the next thing you ought to write is the site of occurrence. Where does it occur? Are you getting my point? That site is now where the liver, particularly the mitochondria of the liver. Then once we are done with that one, the next thing we'll talk about is now the stages. You will now write stages of beta oxidation. Now, like I said in the previous video, there are three stages. And the number one stage, the first stage is called I call it the mobilization of the fatty acid. Mobilization of what? Of fatty acid. So mobilization of the fatty acid is the stage one. While the stage two is called activation through carnitine shuttle. While the third one is now called the main beta oxidation. So these are the three stages involved in beta oxidation. Under this main beta oxidation, that's where you see the four major steps. Those four major steps are what? The first one is oxidation reaction. The second one is hydration reaction. The third one is another oxidation reaction. While this last one is called thiolase. A thiolase reaction or splitting reaction. So basically, thiolase is the enzyme that will do it. So thiolase, okay. Now, like what I just said now is that these are the stages in beta oxidation: mobilization of the fatty acid, activation through carnitine shuttle, and the main beta oxidation. Under this main beta oxidation, that's where you now see oxidation reaction, hydration reaction, another oxidation reaction, and then you see thiolase. I get my point now. So these are the four major steps that you were asked to highlight in the question. Okay. Remember the question said what? Describe beta oxidation of fatty acid, highlighting the four major steps. These are the four major steps. But before you do all this one, you need to give those introduction. You need to have an idea of what it is. Give an introduction, then give the site of occurrence, which is the liver, particularly the mitochondria. You get my point. Then you come to the stages, mobilization of the fatty acid, this and this. So let's start with this first one. Let's not waste time. Let's start with the first one. So the first one there is waiting. Mobilization of the fatty acid. So you write it here. Mobilization of fatty acid. Please don't mind my writing. Mobilization of fatty acid from adipose tissue. Okay. So mobilization of fatty acid from the adipose tissue. Alright. Now the thing is this. Normally the fuel we normally use for breakdown purpose is glucose. Now when the person is in a fasting condition, the body will look for other source of fuel like fatty acid. So let's just say here is our liver. Now. Are you getting my point? This is the liver. Then somewhere here is adipose tissue. I get my point. So adipose tissue, I want to represent it with all these dots. All these dots now in a way are representing fats. You get my point? Then here is the liver. This is the site for the beta oxidation. This is where the fatty acid are stored. So we need to mobilize the fatty acid from this storehouse to the liver to undergo the beta oxidation. So that is what this step one is talking about. I will come again with it. The step one is just telling you that alpha, the fatty acid that you want to use for beta oxidation are stored in where? In this adipose tissue, okay? In this adipose tissue, in this fat tissue. So, and the site where you need the beta oxidation to occur is where? Is the liver. So you need to find a way to mobilize those fatty acid from the adipose tissue to the liver. That is the step, that is the idea behind this thing. And the next thing you will ask yourself is that what is the stimulus for beta oxidation? Like what will make somebody to, in a way, mobilize fatty acid from the adipose tissue? Under what conditions? Like I said, number one condition is usually during so let's say the stimulus. I'm writing something like a stimulus now, or the signal stroke. So let me just say signal. I'm just trying. I hope you get the idea, that will cause the mobilization of fatty acid to this liver. Number one major one is wetin? Fasting condition. Fasting condition. Somebody is fasting, then we will need more energy fuels. The body will seek for other energy fuels like fatty acid. Another condition again that you can think about is what? Which other one can you think about? Let's say somebody is under stress, like in a, let's say the person is being, how do I put it now? It's being is somebody is running, doing exercise or whatever. You get my point now. So the body need energy under stress condition. Your flight and fight response, like when you are under all the sympathetic outflow, you understand? So again, this will be released. Okay. Now under fasting condition, let's say it's because of fasting. That's why we want to actually release the fatty acid that are stored here. Then the hormone of interest will be what? The hormone of interest will be what? It's usually waiting glucagon, okay? It's usually glucagon. That is the one that they usually release, glucagon. Glucagon. While under stress also, we can also release epinephrine. Epinephrine. So hope you are seeing this thing now. So during fasting condition, glucagon is usually released. During stress, we can release epinephrine. Both of these two hormones can act on the adipose tissue to cause the net effect of releasing within fatty acid from this adipose tissue. Then the fatty acid will now be transported in the blood. So let me just put a little pathway here. Let's say this one is the blood now. So the fatty acid will be transported in the blood, going to the waiting, going to the liver. You understand my point? Okay. Let's in a way say what let me say it again. Mobilization of fatty acid from the adipose tissue to the liver simply means the process, the sequence of events that will take place in order for you to mobilize the fatty acid that are stored in the adipose tissue to the liver. What is the stimulus? The stimulus is that maybe the person is in a fasting state. If you are fasting, glucagon will be released, or if you are under stress, then your epinephrine and norepinephrine, which are your sympathetic nervous system, will be what? Activated. You get my point? Both these two can act on this adipose tissue to cause the release of within fatty acid. And you know the form in which fatty acid is stored in this adipose tissue is what? What is the form in which fatty acid is stored here? Shey is tag abi? Triacylglycerol. Triacylglycerol. So please, let's now see the sequence of events that will not take place proper. Now let's see the sequence of events that will take place. Now let's say this is the I want to draw a big cell here. A big cell. Okay, before I draw that cell, where is glucagon released from? Glucagon is released from where? From which cell of the body? From where? Is it beta? Sorry, that would be alpha cell. Sorry, so alpha cells, alpha cell of waiting of the pancreas. You get my point now. Why epinephrine is released from where? From the adrenal gland. Is that not so? Which of the adrenal gland? Is it medulla or cortex? That should be adrenal medulla. So adrenal medulla. So let's actually ship this in. Glucagon from alpha cells of the pancreas, epinephrine from waiting adrenal medulla. You get my point now. All right. So this is the cell now. I want to assume that this one is the cell. What I'm drawing now is the cell in which the fatty acid are being stored. Okay. So let us picture this thing. This is the cell in which the fatty acid are being stored. Then let me just say this one here, under this place here. Here is the fatty acid. Remember the fatty acid are stored as what? As triacylglycerol. So please picture this thing now. Here's our glycerol backbone. That is what I'm shading now. This is our glycerol backbone. Then these are the three acyl. That's why they call it triacylglycerol. These are the three fatty acids, also called acyl for this case now. You get my point now. Alright, three fatty acid esterified to a glycerol backbone. You get my point? So that is how it works, okay? Three fatty acid esterified to a glycerol backbone. That's how it works, okay? This is now what we now call triacylglycerol tag. I get my point. So at the end of the day, at the end of the day, we should be able to find way to mobilize this fatty acid from this place. I get my point. We should be able to find way to mobilize this fatty acid from here. Now, glucagon has been released. When glucagon is released, glucagon will act upon its receptor. You remember glucagon is a hormone, right? It can't enter into the cell. So it will bind to its receptor. Glucagon is a large hormone, cannot penetrate the cell membrane. It will bind to its receptor. This glucagon receptor is usually a serpentine receptor. You get my point? It's a serpentine receptor like this. Attached to this serpentine receptor are some G-coupled proteins. So there are, I believe there are 3 units now. There are 3 units. We have the gamma unit, we have the beta unit, and we have the alpha unit. I get my point. So this is how it works. When glucagon binds to this receptor, Somehow, this alpha unit will leave, will dissociate from that place and activate an enzyme which we call adenylate cyclase. So let's say here on this cell membrane also there's another enzyme here. There's another enzyme here which we call adenylate cyclase AC. Adenylate cyclase. Let me spell that. Adenylate cyclase. I can't spell it. Adenylate cyclase. But this enzyme will be activated. I get my point. So let me come again with the sequence. Glucagon was released. It binds to its receptor. Alpha part of the G-coupled proteins are what is released. The alpha unit is released. It activates adenylate cyclase. Adenylate cyclase will not upon waiting. Will not convert waiting. ATP. It will convert ATP to waiting to cyclic AMP. I get my point. To convert ATP to cyclic AMP. Cyclic AMP now will not do what? Now what will cyclic AMP do? Cyclic AMP now will not convert within protein kinase A. Will not upon protein kinase A. Will activate protein kinase A. So okay, let me just take this one now. This one will act upon protein kinase A. Okay, let's take this one now. Let me cancel this place. So adenylate cyclase convert ATP to cyclic AMP. Cyclic AMP activating protein kinase A. Protein kinase A will now activate another enzyme which we now call the hormone sensitive lipase. Hormone sensitive lipase. Hormone sensitive lipase. In bracket HSL. Hormone sensitive lipase. It is this hormone sensitive lipase that we successfully break down the fatty acid. So we break down the fatty acid and release within 3 fatty acid will be released. 3 fatty acid will be released plus the glycerol backbone will be released. So basically what the hormone sensitive lipase does is that it breaks this bond here. It breaks this bond. It hydrolyzes this bond. Okay. So can I come again what I've said now? I said glucagon was released. When glucagon was released from the alpha cells of the pancreas due to fasting condition, or epinephrine was also released from the adrenal medulla due to what? Stress. Then both of them has the ability to work through this same mechanism. Okay, through the same mechanism. That's how they do work. Okay, now